Hi, and welcome to Chapter 9, Patterns of Inheritance. So far we've learned about different cells, different types of cells, and DNA inside of those cells. But what is your DNA actually doing? That's what we're going to be learning about today. So we know the structure of it, we know that it's kept in our nucleus and it makes us us, but what does that really mean? So the traits that you have are actually traits that your parents had and their parents had, which were passed down. Those traits are known as inherited traits because you inherited them from your parents. Usually when people think of the word inheritance, they think of inheritance, but really inheritance means passing down from one generation to the other, and that's what you're getting. You're getting the genes that were passed down from your parent to you. So that's what we're going to be focusing on today, why you are the way that you are, at least physically. So let's get started with Mendelian genetics. So when we talk about genetics, you really can't talk about it without talking about this guy called Gregor Mendel. And Mendel is known as the father of genetics because he was the first one to really study this, what's called heritable traits. And again, heritable traits is something that's a trait that's passed down from one generation to the other. If you have inherited it, it is heritable, right? It comes from your parents, which came from their parents, etc. So that's why it's called heredity, right? It's something that's passed down from one generation to the next. So genetics is just the study of heredity, right? The study of passing genes from one generation to the next. And nobody did it like this guy, Gregor Mendel. Now this guy Mendel, which you can see right here, was a very famous monk, not famous back in the day, but famous for his research, because it really did give us the first insight into this heredity, this passing down of traits onto the other. I mean, we always knew that brothers looked alike, and we knew that you kind of looked like your parents, but why? That's what Mendel first actually was the one to discover. So I'm not going to show you guys this video, but this is a little biography of Gregor Mendel. I would highly recommend that you guys watch it. We are going to see another video in just a second, which I am going to show you guys. And again, I provide the link for you right down below, so you can always click it and rewatch it later if maybe the quality wasn't so great here on our streaming video. So Mendel did a lot of work on what's called pea plants, right? Pea plants are essentially just that, plants that make, little pea pods. And they're really easy to work with because they are plants, right? They're really quick to grow. They're easy to manipulate, meaning, you can actually get in there and splice a couple plants together. We've been doing that for a while now. And they do come in with a variety of different traits. So you can have yellow peas, you can have green peas, you can have round seeds, you can have bumpy seeds. You can have all these different traits. And that's what he really started to look at because it's too hard to look at people. One, we have a lot of traits going on. We are not as simple as a little pea plant. And two, you can't just look at a simple trait like our color, right? We're not green or yellow, right? We're a blending of all these different things and our environmental factors as well. So he said, I can't look at people, but I can look at plants as a much simplified, what's called a model organism. And then we can extrapolate information off of that because it should all be the same. We're all made of the same DNA, right? Just in a different sequence. So that's what he did. So I'm going to show you this video really quickly right here.
Between 1856 and 1863, Gregor Mendel, an Austrian monk, grew and studied peas. That probably doesn't sound that exciting, but what he learned about genetics changed everything. Let's look at what he did. Mendel bred pea plants, looking for many different traits like color, wrinkly or round seeds, or tall or short plants, among other things. Once he had a purebred trait, he would run an experiment. Mendel crossed two purebred traits. We'll look at green versus yellow colored peas. When the offspring grew, he found that all of the peas were yellow. He couldn't understand why the green trait disappeared. Why didn't the peas have a 50-50 chance of being green or yellow? To explore this further, he took two peas from the first filial, or F1 generation that was just made, and he bred them together. Yes, this is inbreeding, but they're peas, so no one seems to mind. When the second filial generation arrived, F2, 75% of the peas were yellow, but 25% were green. The green peas seem to disappear in the F1, but magically reappear in the F2 generation. What could be happening? Mendel decided that there were factors, which we now call genes. The genes could be either dominant or recessive, and dominant alleles or traits mask recessive alleles. In this case, the yellow is dominant and the green is recessive. So the purebred parents made offspring that had green and yellow alleles, but only the yellow showed because it was dominant. And when the F1 generation bred, there was a chance that they could both give the recessive allele to the offspring, so the green peas showed up again. If you could look at the chromosomes in the pea plants, you would find the gene for seed color. A gene is a section of DNA that controls a trait. The different seed colors have different alleles, which are variations of a gene, like green or yellow. Allele is a fun word to say, and it's actually even more fun to write in cursive, but you can try that next time you're bored. Now you get 2 alleles for every gene, because you have a set of chromosomes from mom and a set of chromosomes from dad, and the same is true for plants. When you write alleles, dominant alleles get capitalized letters, and recessive alleles are lowercase. If a dominant and recessive allele are together, it's called heterozygous. Hetero means different, and the alleles are different, so that makes sense. If the alleles are the same and dominant, we call that homozygous dominant. Homo means same. If the alleles are the same and recessive, we call them homozygous recessive. The letters we choose here are arbitrary, but it's good to use letters that are very different when you view capital and lowercase versions, otherwise it can be confusing. So letters like O are good, but letters like Y, T, or R are great. In pea pods, green is dominant over yellow. If these are the genotypes or allele combinations, what are the phenotypes? Having just one dominant allele of the gene will make it green in this case. So there are 2 green pods and 1 yellow pod. Now this is pretty simple, but let's try thinking about this in terms of making gametes. Assuming tallness is dominant, what would the genotype look like for a man who's homozygous for tall? And what are the possible gamete combinations? Now because tall is dominant, this would be a homozygous dominant genotype, 2 capital letters. Again, you can choose any 2 letters you like, but I'm going to use T for tall. When gametes form, only half of the DNA is in each cell, so only 1 allele can go to each of the sperm that are formed. There's only one possible gamete combination in this case, capital T, the tallness gene. But let's try another case. What would the genotype look like for a woman who's heterozygous for tall? And what are the possible gamete combinations? Now heterozygous means different alleles, so this will be written with a capital and a lowercase t. This also means that there are 2 possible gamete combinations, an egg with a big T for the tallness gene, or an egg with a little t for a short gene. Heterozygous traits will have more variation in their offspring.
Okay, so I hope that video helped. If not, there's plenty of resources online. Just Google it. There's a million videos that hopefully will give you guys a little bit more insights into what Mendel was doing. But I think that was a really good, simple explanation of what's going on. But we'll keep talking about it. So, remember that Mendel is working with these flowers, so they're easily manipulated. Unlike people, which are not, right, we can actually take a white flower, right, a purebred white flower, which in this case would be recessive, and we can take a purple flower, purebred purple flower, and we can combine the 2 artificially. So this is why he did it. This is why he worked on things like flowers and peas, is because they are easily manipulated and he can control exactly what's going on. So just like we saw in the video, we have our parent population, right? In this case, we're crossing a white flower with a purple flower, and in everyone in the F1 generation, or the first generation, right, all of their offspring were purple. And we can actually see this by doing what's called a monohybrid cross, or a Punnett square. Now, Punnett squares aren't everyone's favorite, but they're actually pretty easy if you understand what's going on. Okay, so first off, you draw a box with 4 squares because we're only doing 1 trait in this case. So pretend that up here is your mom, and pretend that down here is going to be your dad. So your mom's side is going to go here, and your dad's side is going to go here. So let's just, in this case, say that the white flower is going to be our mom. So we now know that purple is dominant to white. So if we ever talk about a purebred, that means that you're going to be homozygous. Just like we saw in the video, if you're homozygous dominant, right, that means you're homozygous for purple, right? You have both purple alleles. So in this case, it would be capital P. We're going to use words capital P for dominant, right, meaning purple, and lowercase p for recessive or white. Okay, so if we have a purebred white flower, that means they're going to be homozygous recessive, right? Because purple is dominant over white. So it's the same alleles. For purple would be capital P, capital P. For white would be capital P, lowercase p, lowercase p. Okay, so that's what we're going to write here. So we're going to pretend that mom is going to be the white flower. So she's going to be a little p here and a little p here. Dad is homozygous dominant for purple. So we're going to go capital P here, capital P here. Okay, so this is mom, what's called genotype. And again, genotype, or your genetic makeup. What are your alleles consisting of? Are you capital P, capital P? Are you capital P, lowercase p? Are you lowercase p, lowercase p? Right? That would be that homozygous either. So we have homozygous dominant, capital P, capital P. We have homozygous recessive, lowercase p, lowercase p, and we have what's called heterozygous, capital P, lowercase p. Remember, homozygous, homo means the same, homozygous dominant, they're both dominant. In this case, the capital letter, whatever capital letter you choose. Homozygous recessive, they're the same, but recessive, lowercase, whatever letter you choose. Heterozygous, hetero means different, they are different. You cannot have a heterozygous dominant or a heterozygous recessive. That wouldn't make any sense. You can't have different dominant, right? You would just have the capital P and the lowercase p. Now, when we write this, capital always comes before lowercase, just like in actual English, right? So we would never write lowercase p, capital P. That would be wrong. We would write capital P, lowercase p. That would be how we would write our heterozygote. Now, what we're going to do here is we're going to do a simple monohybrid cross or a Punnett square. So we have mom's DNA over here, we have dad's DNA over here. Now you just have to fill them out. Okay, so let's start with this first box here. We have a capital P here. We have a lowercase p here. Capital P here, we're going this way now. Capital P here. Okay, lowercase p still goes here. Capital P here, lowercase p here. Capital P here, lowercase p here. So in this case, we can see every single one of our offspring is going to be a heterozygote. And that's exactly what you see here. Every single one of them is purple because they're heterozygous. They have one dominant and one recessive. The dominant one masks the other one, and the dominant one is dominant, so it kind of pushes the other one there. He's still here, but he doesn't show up. And this now, we have 100% offspring is purple. Okay, purple is our phenotype. Okay, genotype, genetic makeup, capital P, lowercase p, whatever. Your phenotype, purple, white, bumpy, round, whatever. This is your physical trait. Okay, so these are your genes. Phenotypes are your traits. What do you look like? And this is called gene expression. When it is expressed, when your genotype is expressed, meaning it shows up, that determines your phenotype, right? I have brown hair because my genotype says that I have brown hair, and therefore my phenotype, my physical appearance, is brown hair. Okay? So, we crossed a white flower and a purple flower. Every single one of our offspring came out purple. Hopefully this makes sense to you guys. If it doesn't, try it at home. In fact, it's going to be part of your homework, so try it anyway. Now, imagine, I'm going to erase this. Okay? So exactly what she said in the video, right? They're going to cross again. So in this case, remember these are just plants, it's okay. You're going to cross two of these plants. And yes, they're siblings, but again, they're plants, so nobody cares. Okay, so we're going to cross these two. So what we're doing now is we're crossing a capital P and a lowercase p, right? Remember the heterozygote genotype. Okay, and again, Dad also had a capital P, lowercase p, because everyone did. Everybody was heterozygote. So now in this case, when we cross, we have capital P goes here, capital P. We have lowercase p and a capital P. Remember, we do not write it like this. That's wrong. We write capital letter first. Okay? We go across here, capital P, lowercase p, capital P, lowercase p, lowercase p, lowercase p. So in this case, right, anywhere you see a capital is going to be purple. Anywhere you see capital means it's dominant. It has the dominant trait. The dominant phenotype is going to show up. In this case, we're talking about purple. Now, lowercase p, we don't have any dominant traits, which means he's homozygous recessive. We're now going to get our white flowers back, and that's exactly what we see in the second, oh, I got ahead of myself, in the second generation. If we were to cross those again, if we were to cross the F1 generation with itself, we now get 3/4 purple, right, and 1/4 white. This is a 3-to-1 ratio, right? 3 purple to every 1 white. Okay, so that's what happens in the F2 generation. So this is the F1 generation, the first generation. If we were to cross these guys with each other in the F2 generation, we now see that 1/3 of them come back as those white forms, as the white flowers. This is known as dominance, right? Pure dominance or Mendelian genetics. Straight up dominance. If you find one with a capital letter, it will for sure show the trait. You have to have 2 recessive alleles, meaning 2 lowercase letters, for the actual recessive trait to show up. And we're going to talk about that more in just a second. So here are some of the traits that he looked at, and these are some of the things that he discovered. Purple was dominant to white, meaning white is the recessive trait, the lowercase letter. Right? Yellow is dominant to green. Round is dominant to wrinkles. So he tried all of these different aspects, and he kept noticing that no matter what, if you have one of those dominant traits or those capital letters, no matter what, you're going to have that phenotype, that physical trait show up as the dominant trait. So the recessive ones only ever show up if you have 2 recessive alleles, meaning a homozygous recessive. Otherwise, they are completely lost in the population. They're kind of hiding. It's almost like they're dormant, right? They're carriers of that recessive allele, but they don't show the recessive allele. So their genotype has the recessive allele, but their phenotype does not, right? It doesn't show it. Because, again, all the dominant traits are going to mask the non-dominant recessive traits. Okay, so again, this is exactly what the video showed us. We have homozygous, meaning both of them the same. Homozygous dominant, both capital. Homozygous recessive, both lowercase. Finally, we have heterozygous, meaning 1 and 1. You have 1 dominant trait and 1 recessive trait. And that's because right here, this is, say, mom's gene, this is dad's gene. Remember, they're the same chromosome. We have them on the same, it's chromosome 1 or chromosome 7 or whichever. So they're going to have the same alleles at the same locations. That's what loci means. So anytime you see the word loci, it just means location. Okay, but we're going to have here, if this was mom and this was dad, you can see here, capital P, capital P. The offspring would be homozygous dominant. Lowercase a, the offspring is homozygous recessive. Capital B, lowercase b, now the offspring would be heterozygous. Hopefully that makes sense to you guys. If not, please, please, please practice this at home, and it will. Okay, so again, this is known as just pure dominance, or what's called Mendelian genetics, where the dominant trait is always going to mask the recessive trait. That means this one is purely dominant over the other one, and in the presence of this dominant allele, right, your trait will be masked even if you are a heterozygote and have the gene for it. It's still going to be masked in your phenotype. Your physical appearance is still going to come out purple. So this is known as basically just straight dominance, or also called Mendelian genetics. And that's what we can see in some of these traits for humans. Okay, so we have things like freckles, which is dominant to non-freckles, right? Normally what I would do is I would ask the class, okay, raise your hand if you have freckles. And usually not a lot of people raise their hand, which makes you think, well, if it's a dominant trait, how come more people in the class don't have it? Well, just because it's dominant doesn't mean it's common, right? It just means it's dominant if you have a recessive trait and a non-recessive trait, or sorry, recessive trait and dominant trait, the dominant one will show up. It will just mask the other one. It doesn't mean it's more common in the population. It just means that if it shows up with a recessive one, it will be the one that shows up. It will mask the other one. We have widow's peak, which is dominant to non-widow's peak. Again, not a lot of people have widow's peaks. And we have free earlobes, right, versus attached earlobes. Free earlobes are the dominant one, right? That's the one that you could commonly have. So if you go on to check yourself right now, see if you either have the dominant traits or the recessive traits. It's kind of a fun little thing to do. We usually do it in class, but obviously we're gonna just do it virtually. Okay, so remember that your physical appearance is the phenotype, your genetic makeup, or what alleles you have, either capital, lowercase, whatever, those are your genotypes, your genetic makeup. Phenotypes, physical traits, right? Are you purple? Do you have brown hair? Do you have the widow's peak? Do you have freckles, right? Genotype, are you capital A, capital A, capital A, lowercase a, lowercase a, lowercase a? Meaning, are you homozygous dominant, homozygous recessive, or heterozygous? Okay, so we mix again, we're back to our purple flowers. We have our purple flower mixed with our white flower. So in this case, anytime we say they're purebred, it means they're homozygous. If we're purebred dominant, you're homozygous dominant, capital letter, capital letter. If you're purebred recessive, you are obviously recessive, right? The only time a recessive trait is going to show up is if you have 2 copies of it. So you would have to be homozygous recessive. We cross this. These are our parental units, our P generation, our parent generation. Our F1 generation, or the first generation, comes out all purple. That is that cross that we just did the first time around, where every single one of them ends up heterozygous. When we cross it again, we get these results here, which you can see 1, 2, 3 are purple. 1, 2, 3 are purple. 1 is white. 1 is white. And that's exactly what we saw here in our Punnett square. Now, those are genetic, dominant genetics, but there's also some things called, there's not complete dominance. So if you have something that's incompletely dominant, meaning it's not quite dominant over everything else, so it's not completely dominant. So in this case, what we have here is we have, right, a homozygous dominant red flower. We have a homozygous recessive white flower. When we breed the two, we get a heterozygous, but red was not completely dominant over white. So a little bit of red showed up and a little bit of white showed up, which means we actually get our F1 generation is pink. Excuse me. Let's try to be slick behind the back, didn't work. Okay, so that's what you can see right here. We actually have a pink flower. So in this case, anytime you have incomplete dominance, you have a blending of the 2 traits. We had red, we had white, now we have pink. Okay, so this is known as incompletely dominance. Okay, if it's just straight up dominance, that's Mendelian genetics, it's very simple. This is dominant to this. If you have incomplete dominance, neither one of them is dominance, right? Which means you're gonna have a blending. You have the white mixed with the red, you're gonna get a pink. So if I have a white flower mixed with a black flower, I would have a gray flower, right? A blending of the 2 traits. If we are co-dominant, that means both of them are dominant, right? It's not incompletely dominant, not one of them is dominant over the other, it's co-dominant. They are both dominant. So in this case, we matched our red flower with our white flower and we got a red and white flower because they're both dominant. They're co-dominant. They both show up. So we have parts of the flower that are white, parts of the flower that are red. Same thing over here, parts of the flower that are white, parts of the flower that are red, because they are both dominant and therefore they will both show up in the phenotype. Now, it does get a little trickier than this. There are kind of things like co-dominance, like our blood types. So our blood types are actually co-dominant. Say your mom is a type A and your dad is a type B. That means you are actually going to show up with both, right? A is not dominant to B. They're both going to show up, which means your blood type is going to be AB. So this is something that you can either ask your parents about or find out by kind of just, ask them what their blood type is and then try to figure out your own before you actually ask them what yours is. But, right, you have 2 alleles, one from mom, one from dad. If you are a type A, that means either both of your parents are A, or one of your parents gave you the A gene and the other one gave you it's not the A gene, but it's kind of like the O gene. It means it doesn't have an antigen present. So what's happening here is this is your little red blood cell. And you have these little proteins, these little structures are on the outside. Okay, so these little structures that are on the outside are unique to A, and they're basically like little markers. And so you have these little markers on your red blood cell that says, hey, I'm type A, I only like type A. So if you were to try to get a blood transfusion with, say, type B has very different types of structures on the outside of its red blood cell, which means type A and type B are not the same. Your body's not going to recognize type B. It goes, look, I'm used to these markers. I don't know what these markers are, which means I don't recognize you, and it's going to actually start fighting off that blood as if it were a foreign invader, which is why you have to get a blood transfusion from somebody who has the same blood type as you. Okay, now if you are type AB, meaning one of your alleles was A and one of them was B, you have both markers, which means you can receive blood from type A because you have the marker which recognizes it, or you can receive from type B because you also have that marker that recognizes it. So you can actually get from anybody. However, if you are type O, you have no markers, which means your red blood cell essentially is like naked. It doesn't have any of these little markers on it, which means type O can give to anybody because your body goes, well, I don't recognize anything. There's nothing to recognize. So yeah, come on in. You're totally fine. Which makes type O the universal donor. Because it doesn't have any of those little markers on the outside, it can be accepted by any blood type. Type A, type B, type AB, or type O, which makes O the universal donor. The universal receiver is that AB, because remember, he's got all. He can receive from O because, you know, he has no markers. He can receive from A, and he can receive from B, and he can receive from AB. Okay, so type A is a universal recipient, type B is a universal, type O is a universal donor. I personally have type O blood, which is why American Red Cross calls me all the time to donate. But it's a really, really, really good thing, especially in these crazy times right now. So if you can, please make an appointment. They have very little contact. Blood donation centers still open, and we are experiencing a blood shortage. And don't forget, in all of my classes, I always allow you to do a blood donation for extra credit. If you yourself can't do it, I will allow someone in your family member to do it, but only one time. You can't donate for you and your brother and your sister and your mom and your dad and expect to get all extra credit. No, just one time. But please go out and donate blood because it's really important, especially if you're something like a type O. So again, that's what we were going to hear. We were going to do this in lab. We will probably do something virtually instead because that's what we have to. Okay, we already talked about sickle cell a little bit, but we're going to get back to that in just a second. So, sickle cell is basically where this gene, right, there was a mutation in this one single nucleotide, which switched it from a C to a G, and now all of a sudden we have sickle cell. So our red blood cells are shaped weirdly, right? They're not that big, round, healthy red blood cell shape. Now they're kind of this half crescent moon, you know, sad shape. Unfortunately, it's good for you if you have sickle cell anemia because you won't get malaria, remember? No malaria if you have sickle cell, but it's bad for you because you don't kind of get that blood functioning the way that it should. This is actually known as pleiotropy. Now, pleiotropy basically means where one single gene is affecting multiple traits. So in this case, you don't, so that one single gene is that mutated gene, right, which changes the shape of your red blood cell, right, causing you to have sickle cell, but prevents you from having malaria. So in this case, it controls multiple aspects, right, of multiple aspects of your, I keep touching my face. Don't do that, go wash your hands. It influences several characteristics based on one single gene. So in this case, one gene controls several aspects or several characteristics and traits that you may have. Like, if one gene has sickle cell, you also can't get malaria. So it's multiple based on that one single gene. That is the opposite of polygenic. Polygenic, poly means many, genic means genes. So in this case, something that is polygenic, right, means that many genes are affecting a single trait. And this is where we get things like skin color and height. Okay, so in this case, we're going to be talking about height. And there's about 8 different genes that affect your height. 8 different genes, hence polygenic. Many genes are affecting one trait, your height. So imagine that the white genes are going to make you very, very short, because those are the very short genes. And the black genes, or the very tall genes, are going to make you very, very tall. Okay, most of the population is going to kind of be here in the middle somewhere. You're going to get some white, you're going to get some black, right? You're going to kind of be blended here. So this is what's known as a bell curve, or an average distribution curve, where most people kind of fall under, you know, average height. There's not a ton of people who are really, really short. There's not a ton of people who are really, really tall, right? Because imagine you're flipping a coin, right? You flip it 6 times. You should get roughly 3 heads, roughly 3 tails. Maybe you get 4 heads and 2 tails. But the likelihood of you getting 6 heads and 0 tails is pretty rare, right? Same kind of thing with these polygenic traits. The chances of you getting all 6 genes of the short person gene are going to make you very, very short, but it's rare. You know, the probability of you getting all genes that are going to make you very, very tall are also very rare, right? But it does happen. It absolutely does happen. Something else we have to talk about is what's called sex-linked inheritance, meaning it is on your sex chromosomes. Remember early on we talked about the fact that we have 23 pairs of chromosomes, the 23rd one being your sex chromosomes. You either have 2 X's if you're a female, or you have an X and a Y if you're a male. Well, sometimes those chromosomes can actually tag on these traits. So sometimes they are what's called sex links, meaning you're only going to get them if you have a certain combination of your sex chromosomes. Example, baldness. Maybe you guys have heard that baldness actually comes from the mother's side. Well, here's how this happens. Say the baldness gene is on this little X right here. So she has one normal X, and then she has this gene for the baldness X. Well, the baldness X is a recessive gene, meaning she has a dominant one and a recessive gene, making her heterozygous, but she doesn't show bald. She's a carrier for the bald gene, but she doesn't show it. Now, the husband has a normal X, but he only has one X because he's a man. He has a Y as his other chromosome. So in this case, if we were to do our Punnett square, right, say they have a girl, right, she has a 50-50 chance of being a carrier of this baldness gene. However, because the dad has a normal X and she is a female, which means she gets 2 X's, she gets 1 X from her mom, 1 X from mom, 1 X from dad. She got both of the healthy X's, she's good. She gets the infected X from mom, but she gets the healthy X from dad, so she's still good. She ends up like her mom, a carrier for the baldness gene, but she herself is not bald because it's heterozygous. And she has to have both of the recessive genes to be bald. So in this case, she's okay. So now we look at the male. If he gets the healthy X from the female, great. He's going to get his Y from his dad. That's the only possibility. He has to get the Y from his dad. He turns out just fine. But the other son, he gets the infected X from mom, and now there's only 1 copy of it. He doesn't have a dominant X to kind of hide that other one. The Y doesn't work that way, so he only has 1 recessive allele, which means he's going to be recessive, which means he's bald. So in this case, right, half of the men have a possibility of being bald, but none of the females do. Which is, again, why you're getting it from your mom, but you might not necessarily have it, but your brother or your uncle or somebody, some male in your family might actually have it. Now, I know it's kind of a up-close video, but if anyone cannot see the number in here, it is a number. It is the number 7. Because we're going to start talking about how red-green color blindness is actually one of those recessive traits. Again, it's a sex-linked trait. Unfortunately for you guys, again, we're the carriers for it. We have it on our X's, which means all you guys need is to get one of those infected X's. Infected, it's not infected. It's just one of the X's that carry that color blindness gene, and therefore you guys will be colorblind. So there's color blindness, there's color deficiency. If you can't see the little 7 right here, right, you may be colorblind or color deficient. I am not a doctor. If you're worried about this, go see your doctor. But it is much more common in males than it is for females because, again, we have 2 X's, which means we need both recessive for actually us to show, whereas gentlemen only have the 1, so they really only need the 1 infected X to actually show their baldness or their color blindness gene. Oops. So this is something just to practice on. In this case, we're taking 2 heterozygotes, just like we did up here on the board, and we're crossing them. When it comes to our genotypic ratio, meaning what are the genotypes present, all you have to do is kind of count. Remember, we put dad over here, we put mom up top. Does not matter what order you do them in. Doesn't matter if you know who's mom or who's dad. You're just crossing them. It's going to come out the same anyway because we're looking for the total ratios. So in this case, the genotype, we have one homozygous dominant. These are both the same, right? These are heterozygotes, so we have one homozygous dominant, two heterozygous, and one homozygous recessive. So that would be 25% capital T, homozygous dominant, 50% heterozygous, and 25% homozygous recessive. Our phenotypic ratios, meaning what is physically going to show, in this case we're talking about tallness versus shortness, okay, is 3 to 1. Because there's a capital letter here, there's a capital letter here, and a capital letter here, which means all of these are now going to show the dominant trait. The dominant trait is tallness. You're going to have 3 that are tall, or 75% that are tall, and only one recessive, homozygous recessive, meaning only one that actually comes out short. Okay, you guys will have a homework assignment due online on the Canvas site, so make sure you guys do this and actively practice this. If you have any questions, rewatch this video, or you can Google any number of resources online. I think Crash Course even has a Mendelian Genetics video online. Please go watch it. It really, really does help. And I wish you guys the best of luck, and I will see you next week.